Hello dear students, welcome back to your science class. Today I am going to continue with the topic cell. In our previous lesson we have learned about definition of cell as a structural and functional unit of a body, cell theory, discovery of cell, number, shape and size of the cell. Today we are going to learn structure of a cell. So let's start. If we study a cell under microscope, we would come across three features in almost every cell. What are those? Plasma membrane, nucleus and cytoplasm. All activities inside the cell and interactions of the cell with its environment are possible due to these features, due to presence of this plasma membrane, nucleus and cytoplasm. Inside the cytoplasm, there are different cell organelles performing specific functions. What are those cell organelles? They are endoplasmic reticulum, Golgi body, mitochondria, lysosome, vacuoles, centrosome. So, let's learn about them in detail. First, plasma membrane or cell membrane. As you can see the structure of the plasma membrane, it is made up of lipid bilayer, two layers of lipid. And there are some proteins embedded into this lipid bilayer, and fats are also present in the structure of the plasma membrane. So we can see cholesterol, glycolipid, etc. in the form of fat. So this is the structure of a plasma membrane or cell membrane. Extremely delicate, thin, elastic, living and semi-permeable membrane. What does it mean? Semi-permeable? Semi-permeable means selectively permeable. Plasma membrane or cell membrane allows certain substances to enter through the cell while prevent others to enter. That is why it is known as semi-permeable. Made up of two layers of lipid molecule in which protein molecules are floating. That's why this model of plasma membrane known as fluid mosaic model can be observed under an electron microscope only, cannot be seen with simple microscope, even cannot be seen with compound microscope. What is the function of plasma membrane? Maintains shape and size of the cell, protects internal contents of the cell, regulates entry and exit of substances in and out of the cell. That is why it is semi-permeable. It can regulate entry and exit of the substances inside a cell. Cell wall. Outside the cell you can see cell wall that is present in plant cell and bacterial cell, absent in animal cells. We will learn about their features. Non-living and outermost covering of a cell. Plants and bacteria. As I already told you, they are absent in animals. Can be tough, rigid and sometimes flexible. Made up of cellulose, hemicellulose and pectin. May be thin or thick, multi-layered structure. What are the functions? Provides definite shape, strength and rigidity to the cell. In case of animals, we have skeletal system to protect the internal delicate organ. Plants do not. So cell wall provides them with shape, strength and rigidity. Prevents drying up of cells. Helps in controlling cell expansion. Give it a definite shape. Protect cell from external pathogens so that they cannot enter into the cell easily. So these are the function of cell wall. Next is nucleus, most important part of the cell. You can say it is as a controlling part of the cell. Okay. Dense spherical body located near the center of the cell. Diameters varies from 10 to 25 micrometer. Present in all cells except red blood cells and sieve tube cells. Red blood cells present in human body we all know, and sieve tube cells present in plants. So why they are absent in red blood cell? We know the function of the red blood cell is to carry oxygen and to supply oxygen to all the parts. If red blood cell contain nucleus, it itself utilize some of the oxygens. So it do not have nucleus. I will mention you mature red blood cell lacking nucleus. Okay. So red blood cell and sieve tube cell in plants, they do not have nucleus. Well developed in plants and animal cells. Underdeveloped in bacteria and blue-green algae. We will discuss about detail about this in next portion of this video. Most of the cells are uninucleated, having only one nucleus. Few types of cells have more than one nucleus, as in our skeletal muscle cells. Structure of a nucleus. What are the parts present in a nucleus? A true nucleus contains the following parts. Nuclear membrane that covers the nucleus. Nucleolus present in the center of the nucleus. Nucleoplasm, that is the sap-like thing that is present inside the nucleus. And chromatin fibers. These fibers condense to form chromosome during cell division. Chromatin fibers you can see. So a true nucleus contain nuclear membrane, nucleolus, nucleoplasm and chromatin fiber. Functions. Control all the activities like metabolism, protein synthesis, growth and cell division. Nucleoli synthesize ribonucleic acid RNA to constitute ribosome. Store hereditary informations in genes. That is why I told you that nucleus is the control center of the cell. Next is cytoplasm. We have discussed in our previous video that nucleus and cytoplasm together form the protoplasm. They are the living part inside the cell. What is cytoplasm? Jelly-like material formed by 80% of water. Present between the plasma membrane and nucleus. You can see this is the cytoplasm that is present between nucleus and plasma membrane. Contains a clear liquid portion called cytosol. Also contains many organelles with distinct structure and function. So now it is the first cell organelle we are discussing about, endoplasmic reticulum. You can see it is endoplasmic reticulum. Network of tubular and vesicle structures which are interconnected with one another. Two types of endoplasmic reticulum are there. Smooth endoplasmic reticulum, that is lac ribosome, abbreviation is SER. And rough endoplasmic reticulum starred with ribosome, abbreviation is RER. You can see rough endoplasmic reticulum while ribosome are embedded in the endoplasmic reticulum, and smooth endoplasmic reticulums are devoid of any ribosome. What is the function? Gives internal support to the cytoplasm. Rough endoplasmic reticulum synthesize secretory proteins and membrane proteins. Smooth endoplasmic reticulum synthesizes lipids for cell membrane. This is their function. Next is Golgi body. Got its name from the discoverer Camillo Golgi. This is the structure of the Golgi body or Golgi body. The face, that is facing the nucleus is called cis phase, and opposite phase is known as trans phase of Golgi body, formed by stacks of 5 to 8 membranous sacs. In plant cells, Golgi body is not that well developed and it is known as dictyosome. What is the function? Modifies, sorts and packs materials synthesized in the cells. So its function is something packing and storing and sending. Okay. Delivers synthesized materials to various targets inside the cell and outside the cell. Secretes different important enzymes and proteins. Lysosome, interestingly known as suicidal bag. They look like small spherical single membrane sac. Most of the cell organelles are double membraned. Lysosome is single membrane sac. Found throughout the cytoplasm, filled with hydrolytic enzymes. Occurs in most animal cells. What are the functions? Helps in digestion of large molecules. Protect cell by destroying foreign invader like bacteria and virus. So bacteria and virus enter into the cell, are destroyed by these hydrolytic enzymes. Degradation of worn out organelles. In dead cell, perform autolysis. That means if any cell is dead, then lysis degrades its own cell. That is why it is known as suicidal bag, because it is having the power of degrading the cell inside which it exists. So it is known as suicidal bag. Next, vacuoles. You can see in plant cell a large vacuole that is very prominent, whereas in animal cells small vacuoles but large in numbers. Single membrane sac filled with liquid or sap. That sap contains water, sugar and ions. In animal cells, vacuoles are temporary, small in size and few in number. In plant cells, vacuoles are large and more in number. Just now we have compared between the two pictures. Large vacuoles and smaller and more in number. Function: store various substances including waste products. Store food particles in amoeba cell. You have learned about amoeba food particles. Provide turgidity and rigidity to the plant cell so that plant cell can maintain its proper shape. Mitochondria, the powerhouse of the cell, is very important cell organelle. Small rod-shaped organelles bounded by two membranes, inner and outer. Contains their own DNA, which are responsible for many enzymatic activity. That is why it was told that mitochondria is able to maintain its autonomous existence as it has its own DNA. Function: synthesize energy-rich compound ATP, adenosine triphosphate. This is the energy currency of the cell. I repeat, adenosine triphosphate. As because mitochondria synthesize ATP, that is why it is known as the powerhouse of the cell. ATP molecules provide energy for the vital activity of living cells. Plastids. Plastids are double membrane bound organelles found inside the plant and some algae. This is absent in animals. They are responsible for activities related to making and storing food. They often contain different types of pigments that can change the color of the cell. There are three types of plastids, namely chromoplast, they are the colored plastids, chloroplast, green plastids, and leucoplast, white plastids. Let us know about these plastids a little in detail. Leucoplast. Leucoplasts are colorless plastids that store foods. They are found in storage organs such as fruits, tubers and seeds, white in color, store foods. Chloroplast. Double membrane bound organelles found mainly in plant cell. Inside the chloroplast, chlorophyll is present. Convert light energy into chemical energy in the form of food. So this is the function of the chloroplast. Provide green color to the leaves, stems and vegetables. Chromoplast, the color plastids. Chromoplast are plastids that produce and store pigments. Different colored pigment they can contain, namely carotene, xanthophylls, etc. They are responsible for different colors found in leaves, fruits, flowers and vegetables. Next is centrosome. This is the structure of the centrosome. Inside the centrosome, two centrioles are present. They form almost 90-degree angle with each other, and these are the microtubules. Centrosome is the membrane-bound organelle present near the nucleus. Consists of two structures called centrioles. Centrioles are hollow, cylindrical structure. Centrioles are arranged at right angle to each other. Function: form spindle fibers which help in the movement of chromosomes during cell division. So they help in the process of cell division. Now we will learn about difference between plant and animal cells. You have learned in this video that certain cell organelles are present in plant cell, certain cell organelles are present in animal cell. We will quickly take a look at the difference between plant and animal cell. Having plastid, of course, they contain chloroplast, that is why they are able to perform photosynthesis, do not have plastid. Have a cell wall to provide its rigidity so that it can protect itself from change in different environmental conditions. Do not have cell wall. Animals do not need it because they are capable of movement. They can move somewhere else if the environment is not suitable for them. Have a large central vacuole in plant cell. Have small temporary vacuoles if any. May have plasmodesmata, that is the connection between the two cells through cell membrane. Do not have plasmodesmata. Do not have centrioles in plant cell. Have paired centrioles within centrosome in animal cell. Plant cell do not have cholesterol in cell membrane. Animal cell have cholesterol in their cell membrane. Store excess glucose as starch, and animals store excess glucose as glycogen. Generally have a fixed regular shape in plant cell due to presence of cell wall. Generally have an amorphous shape, as you can see in case of amoeba, no definite shape. Now another interesting and important thing, that is difference between prokaryotic and eukaryotic cell. As the name suggests, prokaryotic cell, pro means primitive, karyon means nucleus. Prokaryotes contain primitive nucleus. Their nucleus do not contain any nuclear membrane. Rather, nuclear material present in somewhat center of the cell, and that is known as nucleoid. They are having cell wall, unlike animal cell. Whereas eukaryote, as the name suggests, eu means true, karyon is nucleus, where true membrane bound nucleus is present and different membrane bound cell organelles are present, which is lacking in prokaryotes. So this is the basic difference between prokaryotic and eukaryotic cell. So in this lesson, we have mainly learned about all the parts of a cell, structure of a cell, different cell organelles and their specific function. We also have learned about difference between animal and plant cell, and we have learned about difference between prokaryotic and eukaryotic cell. I hope your understanding about this is clear now. If you have missed the first video of cell, then please watch the first video first and then see to this. Thank you.